So now I request Dr. Poonam Misra Madam to formally start this session. Madam.
Yes, sir. Thank you so much, Gopalji, sir. And I welcome all the participants to this online platform for the Science Dialogue series, as well as our today's speaker, Professor Abhijit Majumder, sir. Before I introduce all of you to our today's speaker, Professor Abhijit Majumder, I would like to give a brief introduction about this Science Dialogue series, since many of the participants have been joining us from probably from last 6 months or from 1 year. So many of them would be knowing about the Science Dialogue series, and to those who are new to us and join us for the first session or from the last 1 or 2 session, so for them this Science Dialogue series is basically a brainchild of Gopalji sir, who started this particular series for the benefit of the students, as well as uh, for the benefit of the scientific community, by bringing all the persons who are having expertise into different domains of the life sciences, so that all of us can get benefited from the experience of all these distinguished guests who are appearing on this platform. So in that continuation, today we have Professor Abhijit Majumdar sir. So a warm welcome to Professor Abhijit Majumdar, and to our participants, just a brief introduction about the Professor Majumdar, who is a distinguished faculty member in the Department of Chemical Engineering at IIT Mumbai, with a research portfolio that bridges cell mechanics, microfluidics, and stem cell biology. If we talk about his academic journey, that includes a PhD from IIT Kanpur, followed by a postdoctoral research at the ISTEM Bangalore and Harvard Medical School, Boston. His work focuses on how mechanical signals influence the behavior of stem cell. He investigates how cellular morphology, contractility, and mechanoresponse are interconnected, aiming to uncover universal predictors of the mechanosignaling. His lab at IIT Mumbai is at the forefront of the mechanobiology, exploring how physical forces and mechanical properties of the cellular environment influence the stem cell fate. He is also pioneering the use of microfluidic platform to stimulate complex environment where both chemical and mechanical gradients can affect the cell migration as well as their differentiation. If we talk about rest of the things, so beyond the lab, Professor Mazumdar is passionate about the outreach of the science, particularly among the students. He is a well-recognized personality with the various prestigious awards, including the TR35 Indian Young Innovators Award by MIT Technology Review and the DBT Wellcome Trust Early Career Fellowship. Moreover, he has more than 50 publications, research articles in the form of either book chapter or the original research article. So he is a very active person in the field of research related to the microfluidic platforms in the stem cell biology, having more than citations of 1,000. So I welcome Professor Majumdar to this platform, and uh, expect that our participants will also benefit from your huge experience in the field of stem cell biology connecting with the microfluidics. So, sir, we welcome you for this platform,
And
and we hope.
most
Yes,
important thing is he has 30,000 followers on Twitter X. So you
yeah,
can.
he's a social media influencer, kind of in
Yes,
scientific
yes,
community.
yes. So
Yeah.
generally there is rare mixture कि scientists are also influencing. But हमारा science dialogue series का यही purpose है. Scientists should be celebrity, not the dancer, singer and all है ना जो jokers. Scientists and professors should be model for the society. So whatever we can do all these things. And we are grateful to you that you are accepting our invitation and ready to deliver lecture. And this topic is also very important.
Yes, it is organ on chip, a future technology. So I think
Yes.
this would interest our participants too. So, sir,
Now,
we
sir,
can have.
platform is yours.
Yeah, platform is yours, and
Okay, so thank you, Gopalji sir, and I think this is a very, very interesting platform that you have created. Um, Sorry, this is not the one. Sorry. Yes. So Yeah, so it is a, so my screen is visible?
Yes, yes, visible,
Yes,
sir.
great. Yeah, thank you. So yeah, so you have created a fantastic platform, and as you have just said that scientists should be celebrities. I actually kind of agree with you, on that part, that not really the scientists to be the celebrities, but the overall, we should celebrate the science. I really, really strongly a believer of that. And in fact, I often ask this question to multiple people, that if I ask, anyone to tell the name of five Bollywood superstars or five cricketers, five footballers, five tennis stars, etc., living, who are right now, people can tell about not just the Bollywood stars. They can tell about their son, their father, their daughter, whether their son has a good potty today morning or not. Everything people knows. But at the same time, if you ask a common person that name five living Indian scientists, not who are no more with us, but five living Indian scientists, often we don't know. And that is not the failure of the common people, but I strongly believe that that is failure from our side, that we could not go beyond our lab, beyond our institute, and to reach the common mass. So in that aspect, I think you are doing a fantastic job, and thank you very much for
More
giving me.
than 120 lectures or free lecture has been organized, sir.
So that is like fantastic. That is really, really, really amazing job. And Poonam madam has given a really long intro about me. I mean, I can see that she has actually gathered all the informations about me. So thank you, ma'am, for this time.
Madam is working at Baroda, KPGU. She is helping me in this endeavor.
Right. Yes, thank you, ma'am. So let us jump into our today's topic, and our today's topic is a futuristic idea. It is, the technology is not something that is right now here, but it is being developed, and it is called organ on chip. So as I have kind of tried to put a cartoon here, that why the laser is not working? Where is the laser? Huh. So as I have kind of put here, that we have a organ, say like a heart, and then on a chip. But in the next kind of 40-45 minutes, we will try to understand that why do we need organ on chip, what is organ on chip, how they are made, and then what is the future. So some of these things we will try to understand. So the first question is, why do I need an organ on chip model? So before going there, let me tell you that how the drugs are discovered. Probably many of you know, but just for the general audience, say for example, we know that if I fever, I need to take a paracetamol. But how do we came to know that paracetamol work in fever? Or say if I have common cold, vitamin C works or it does not work. So how do we get to know that? So that essentially happens through something as known as drug discovery, and there are the various parts or phases of this drug discovery pathway. So drug discovery pathway, it starts with taking a 5,000 to 10,000 possible compounds, that is generally computer generated. So we call it in silico model, and using these 10,000 compounds, we will try to look at that, okay, if this is a particular target, for a particular disease, which molecule can block the receptor, can interfere with the pathways, something like that. So that is what we start with, the 5,000 to 10,000 compounds. After doing this in silico computational analysis, which may go up to 3 to 5 years, finally we bring it down to say 250 compounds. So these numbers are just representative numbers. So say 200, 250 compounds. Now comes the section that is called the preclinical trial, where this organ on chip will come into picture. So what we do in the preclinical trial is that we do this in vitro study where we have the cell culture plate, we grow the cells, and then we add our, whatever drug we would like to use, we add that, and we test the effect. And then we go to the animal studies with the small animal, with the mouse, or then, depending on the drug, we can go to rabbit, even in the, the chimpanzee, or, in a, into the higher order mammals. But the broad idea here is that then we check in the animal for drug safety, drug efficacy, and then how the drug is distributed into various organs. Now this may go for 1 to 2 years. There, after that we start the clinical trial where we work with human. And there comes the interesting part of the story. Now you think of that this total preclinical trial, that is in vitro as well as in animal, in vivo, that is a very stringent check, okay, both for efficacy and toxicity or safety. After a very stringent check, so you can think of it as an, at an exam, that a very stringent exam goes on, and out of that, say here out of many drugs, say if I pass here from that exam, say 100 drug molecules that will pass this preclinical exam. Meaning is that I am believing that these 100 molecules are good enough to be tested in the next phase. But when we go to the next phase in the clinical trial, what happens? There happens the disaster that out of those 10 molecules or compounds which passed the preclinical test, 90 or 95 of them fail in the clinical trial. Now that is very, very important part, right? That you chosen 100 drug, you went for clinical trial, and clinical trial go for 6 to 7 years, and it is very, very expensive, and where the human lives are involved. And what is happening? That chosen, and I am kind of stressing upon this point again and again, that these 100 are not arbitrarily selected molecule. These are the chosen one. And out of even this chosen 100, 90 or 295 fail. That tells me somewhere that the testing that I am doing here is not good enough. It is not being able to predict that what is going to happen when I will use these drugs in human. So I'm testing in animal, but when I'm trying to extrapolate that result to human, that extrapolation fails. Now you think of it from the another angle. Suppose 100 times I take train to go somewhere, and out of that 100 times, 90 times, it takes me to somewhere else. Will I take the train ever? Probably not, because it is absolutely unreliable. उसके ऊपर भरोसा किया नहीं जा सकता है. But for decades, we are trusting on this particular way of doing the preclinical testing, which has a success rate in clinical trial is less than 10%. So this is really a big, thing to think of. What it leads to? It leads to a really long, almost 20 years, 15 to 20 years for a new drug to come to market. In this time period, many patients are dying. Real people are dying because the drugs are not there. And drugs are not there because the testing is long. And the testing is long because our predictive ability in the preclinical trial is bad. Also, it increases the expenditure, the cost of a drug discovery enormously high. And there is another point which people rarely talk about, and I think that is also a very important thing to discuss, that I started with say 1,000 compounds here. After this testing, I took only 100 compounds for the clinical trial. The all 800 I actually discarded based on my in vitro or in vivo result. Those 800 molecules never ever got a chance to come to the clinical trial. Now, if my prediction is so bad to predict which molecules will be good in the clinical trial, what if my prediction is also bad to tell which molecules are not good for the clinical trial? Meaning I have discarded these 800 or 900 compounds, and once that data will be there, nobody else will test these 900 compounds for the same disease. So are we losing very important potential drugs in this process? We don't know. So why this is happening? This is happening. So this is I have already discussed. So I'll just keep that part. This is happening because in our body, life is very complex. Our cells, they don't live in our body as a separate thing. They are like a social animal. They interact with each other. There are various different cell types. There are the neighboring cells. There are the soluble factors. There are signaling process happening all the time. Some are the physical signal. Some are the chemical signal. All of those things happening. When I do our testing in the lab in this kind of plates, it's a very different environment. Now, if I give you an analogy, when I am talking to you, sitting here in this online platform, and once the meeting will be over and I will go home, will I behave the same? No. Here the way I am behaving and the way I will behave in my home, in front of my parents, I will behave in a certain way. With my friends, I will behave in a different way. With my students, I will behave in a different way. So our behavior is very much dependent on our environment. Similarly, how the cells will respond to a particular drug molecule, that depends on its environment. And the environment where I am testing them is nothing or nowhere close compared to what they experience inside the body. You may argue that, okay, but we are testing in mouse. That is a full body, it has all the signals. Yes, you are right, it has all the signal. But a human is not a 60 kg mouse. Our genomes are different, our metabolomics are different, our microbiomes are different. We are not the same. So that the testing result done in a mouse cannot be directly extrapolated to human, and that's why the clinical trial fails. So there comes this futuristic approach, which is called the organ on chip. The idea is to bridge the gap between the in vitro versus in vivo, and can I kind of bridge the gap between my preclinical to clinical, or what I test to what I expect to see in human. So that is the idea of the organ on chip. So what is organ on chip? So as I was saying is that so the cells have the various different environment that they experience inside our body. Is the organ on chip something that we would like to mimic a human organ in every aspect? The answer is no, because we are not creating an artificial organ. So if I say a heart on chip, so that is something everyone needs to remember very clearly, is that if I say I'm making a heart on chip, that does not mean we are making an artificial heart that can be implanted in a human body. No, we are creating a system which will resemble the human organ, so that it can be used for an artificial testing platform. I will elaborate on this a bit more here now. So when I say the resembles, what is the meaning? The first is that we will always use the human cells, because remember, we are trying to mimic the human body, or we are trying to mimic the response that we will get in human. So we will always use the human cells. And generally we use more than one cell types because we would like to know the cell crosstalk. Generally they are done in a three-dimensional manner. So our regular cell culture plates, which are generally 2D, two-dimensional, here we talk about three-dimensional structures. Then what I will call as an organ on chip, it must capture at least one structural aspect and one functional aspect at least. So I will explain that with further examples. But it should say, if I want to mimic heart, it should at least mimic one structural aspect of heart and one functional aspect of heart. And generally we have some kind of microfluidic flow in our system. So it may look like, something like this, as you can see here. So by microfluidic channel, I mean that these are the basically hollow channels. These are the openings. So you can have your media passing through. These are opening. So unlike our static culture, where we do not change the cell media, cells are sitting in the same media. In case of this organ on chip, there is always a flow, again mimicking the body, so that things are, new nutrients are coming, and what cells are secreting, going. We are taking it off depending on, again, the flow rate that we want to mimic. These channel diameters are often like your, like one of your hair. It is that thin. That also reduces the cell, the number of cell required or the amount of reagent required, etc. Also it helps. So it looks something like this. So essentially we can have the multiple cell types and in some kind of structure. So do we have any, so we take a question, intermittently or at the end? I
Yes.
generally prefer.
सारे लोगों को अनम्यूट मोड में है. If they think it necessary to ask, they can unmute and ask. Otherwise, after lecture, they will ask.
So up to this point, if anyone has any question, because
Sakshi
now,
Shah, you have raised hand. Do you have any questions, Sakshi?
Yes, sir.
yeah, please
हाँ,
go ahead.
पूछिए.
Sir, I want to ask, is there any difference between organoid or organ on chip? Is there any
Okay.
difference?
Very good question. I will answer it, but just let me quickly check if there is any other question. Anything else?
No question, sir.
Okay. So Sakshi, it is a very good question. I actually generally put that. Today I have not put it. Organoid is generally again a three-dimensional structure that generally starts with some kind of stem cells, and it is a three-dimensional, like a spheroid, like a ball, and then they differentiate to create different cell types in the same structure. So the purpose is again very similar for the testing, research, drug testing, etc. But generally, organoids do not have such kind of complexity in terms of their design. It is generally just like a three-dimensional, the collection of cells.
So organ on chip doesn't involve any kind of stem cells.
It can. So the organ on chip can involve the stem cells, it can involve organoids, etc. But organoid will not have this kind of microfluidic flow or this kind of geometric structure.
Okay, sir.
So basically this organoid, organ on chip, they all come under a bigger umbrella that we call as microphysiological system. So we would like to mimic the physiology in a micro scale. So they come under the same family, but they are the cousins, but they are not the same.
So, sir, does what you said about perfusion or microfluidic flow, does they help in some clinical trial?
I will show you one example, you will get to know. Okay, please just hold on. Okay, is there any other question?
No, sir. Please continue,
Okay.
sir.
So when we talk about the organ on chip, the one question I was kind of trying to answer is who first made, at least as we have the documented, the first documented organ on chip. So the organ on chip technology, as we said, it is a very new thing.
One
So it's,
question written in chat. What material it is?
oh, so this material is called PDMS, polydimethyl siloxane. It is a kind of a polymer. It starts with a liquid and then it solidifies into the structure.
Yes, please continue, sir.
Okay. So this is a very recent field, recent development, and somehow around 2010, 2012, this first development of this organ on chip started. However, if I stretch it a little bit, the story goes, I could at least reach somewhere between 1452 to 1519. So the person who made this drawing, that this was this person's lifespan. Can anyone tell me this drawing is by whom? Does anyone know? Okay. Now do you know whom I am talking about?
Leonardo.
Absolutely, yes. So Leonardo da Vinci, whom we know as an artist, and this is the most famous art form, but I would say he was a scientist, because here you can see that he has made a detailed diagram, and there are many more fantastic diagrams if you look at. He made the detailed diagram of human vasculature system. And remember, at that point of time, there was no MRI, no X-ray, nothing. People even did not know that the cell exists. And at the time, he made this whole thing. So how he made this structure? And that comes in my opinion, that is the first organ on chip. So what he did, he exhumed a human body, which was not approved by church at that point of time, but he exhumed the human body. Then he put paraffin in the blood vessel. So you got the paraffin structure of the blood vessel. Then he embedded that structure in a transparent polymer, which is essentially our today's PDMS. I mean, he did not use PDMS, but it resembles what we are doing now. So he embedded everything in a transparent structure. And after embedding the transparent structure, he heated it so that the paraffin got molten and came out as a liquid. So what he got is the hollow channel that you see here. So he got this whole hollow channel vasculature structure. Then he had water flow through those structure to see the blood flow, like my microfluidic perfusion. And because he did not have the cell, but somehow he thought that this blood is not just liquid, it may have some particle or corpuscle. I have no clue why he thought or from where he got the idea. But to incorporate that, what he did was he used some seeds like chia seeds and mixed it with the water. And then he observed the flow, that when those seeds flow through that and when they come to a bifurcation of a channel or some kind of turns and twist, how the flow takes place. So essentially, in my opinion, that what he did was he created an organ on chip to understand the blood flow. And as now I urge you to connect that one structural aspect, that okay, it is a vasculature structure. One functional aspect, blood flow. And he combined these two things to observe something. So from there, I now jump many centuries and I come to one of the very first organ on chip, that is called lung on chip, that was made in the Harvard Medical School. and Wise Institute. And now there is a company called Emulate. They have kind of using it. So what is here, I first will show you just quickly, and then I will show you a video. So there is a membrane here. Then one side you have lung cells. Another side you have blood vessel capillary cells. From this lower channel you are flowing the blood, upper channel you are flowing the air. So basically you are mimicking one alveoli structure, alveoli wall, or your lung has this alveoli, right? That structure of the wall through which the oxygen or carbon dioxide transport takes place. And then we will see the video what happens. So let me just go to the video. Sorry. So please let me know if my video is visible. Can you see my video?
Yes, it's visible. Yes, sir, visible.
Okay, please. Yeah. So when we breathe, what happens is that the lung expand and contract, expand and so we have a sinusoidal motion here, and then we have the blood flow, right? Blood and the air flow. So that is what they are trying to mimic here. So this is a chip which looks like a computer memory stick, as they say. And then you have this membrane here. So in this case, this membrane is also made of PDMS, but you can use other membranes also. And this membrane has lots of small pores here. One side you have the lung cells, another side you have the capillary cells. So it is basically mimicking the lung structure. And then these vacuum channels are created to create this sinusoidal stretching and relaxation mode to mimic the functionality of the lung. So top part is my air channel flowing air, bottom part is the blood channel flowing the media. And then what they did in the top part, they are flowing some respiratory bacteria, which will cause a respiratory infection. And in the bottom channel, they have the white blood cells. Now, when a bacteria get attached to the lung epithelial cells, that sends out the signal so that the white blood cell crosses the barrier and kills the bacteria. That is what they are trying to mimic here. So what now you are seeing a real-time video, and you can see the white blood cells are moving when there is no bacteria. So they are just flowing. As soon as they will introduce the bacteria, you can see that they are now getting engaged. They are getting immobilized. They are not flowing anymore. Some are flowing, but some are now getting immobilized. So this is one which will immobilize and will try to come through this hole, but it is going out of focus, so you will not be able to see. Now you can see again that where you can see that they are kind of coming in the ER channel from the blood channel. And now this is a real-time video again. The green are the bacteria, so red blood cells. They came from the blood channel, crossed the epithelial endothelial barrier, came to the other side, and then killed the bacteria. Okay, so I'll stop here now this video. Okay, so then again here the whole idea is that, so what was my one structure? So okay, let us go back how I defined the lung on tube. Number one, human cells. Yes, I have used human cells. Have I used more than one cell type? Yes, I have the lung cells and I have the capillary cells. Have I used one functionality? Actually, I have used, before function, have I used one structural aspect? Yes, this is the one structural aspect, that I have a membrane mimicking the basal membrane in our lung. I have one side, I have the lung cells. other side I have the capillary cells. Then what function I have mimicked? I have mimicked the function of this sinusoidal stretching. And this is not just the one thing. Then they have introduced the disease, the inflammatory disease, introducing IL-2. And in the inflammatory disease, the lung accumulate fluid that also they could replicate in their model. Okay, so then there are various other models. One is, say, for example, tumor on chip, where I have now again very similar kind of structure in that way, that I have a membrane. One side I have say I'm trying to mimic the breast cancer. So I'm having the mammary epithelial cells. Then I have my tumor cells here. Then I have my stroma, that is the the stromal layer, where I may have the fibroblast. So this is basically to mimic that how the metastasis takes place, that how the tumor cells may cross the stromal barrier, endothelial barrier, reaches to the blood vessel, then reaches to some other region where they further again cross the blood vessel barrier, stromal barrier, and repopulate the tumor. So whole the metastasis process that can be recapitulated in this kind of tumor on chip model. So something this is just one slide that we are showing from our work. But before again going there, is there any question up to this point? That for the lung on chip and the tumor on chip part.
One question has been asked: is there any other material that can be used out here than PDMS? And if so, what are the main things to keep in mind for a selection of the material? Evelyn D'Souza.
Thank you for this question. I actually removed that slide. I thought that slide might not be interesting for the bio background people, but I am sure my assumption was completely wrong. So let me just try to find out. So the whole how part I kind of removed, but looked like people have. So why we are using PDMS? So PDMS, number one, it is non-toxic, it is non-cytotoxic, it is basically the silicone which used as an implant to enhance our body in the cosmetic surgery. Then they are air permeable, so actually I should write here they are gas permeable because they allow the gas to permeate. Then, as you have already seen, they are transparent, so easy for the real-time microscopy. They are soft and deformable, so to have this kind of stretching kind of application, they are very useful. And because we know how to attach one PDMS layer with other, so we can use a multi-layered structure very easily. So that's why we use PDMS. Now, to answer your question, yes, many other materials can be used. So various different polymers like the polystyrene, polymethyl methacrylate, acrylic. So a lot of the other materials can be used. Then we can use the different types of extracellular matrix, gels. We can use glass, just etching the glass and use that as your organ on chip model. But that it may not have all the good properties that PDMS has. For example, one of the major bottleneck using other polymer is that often they are not gas permeable. So if you have a flow, that is fine, but if you don't have a flow, then the cells may not get enough oxygen, for their growth. So yes, so the short answer to your question, there are other polymers to be used, but they should have all these good qualities ideally. Any other question?
Yes, one more question.
Good evening, sir. I'm Dr. Sandeep Kumar from Parul University. I have just started in this area to to focus to PDMS microfluidics. So actually, I want to incorporate peptide. So is there possible in the microvesic that channels created by the machine? So is there possibility how we can adherent that our peptide in the channels?
So you want to deposit your peptide on PDMS layer, right?
Yeah, that channels created inside the PDMS. So I want to deposit inside the channel.
Yes. So that is possible. You can. So the peptide or the protein deposition is absolutely possible. You need to do some surface modification, and
Yeah,
then
that
those things.
because I am not expertise, so that's why I need your help. So how can we?
So I
Dr. Kumar,
request to discuss more in more
exactly.
detail after lecture.
So
Just
that
Yeah,
is
yeah.
what I was coming
just
to that
stick
point.
to the question which he has delivered. After
Yes, sir.
finishing the lecture, you can get more discussion and cross questioning. Sandeep ji,
Okay, sir.
please wait.
Yes. Okay, sir.
Yes,
Sure.
please continue, sir.
Okay. So now I will just give only one slide to tell you one thing that we are working. So this work we are working with Dr. Deepak Modi from ICMR NIRRCH, and his student Anshul and my student Saurabh, they are working on it. So here we are talking about placenta on chip, right? So what is placenta? So as you know, placenta is a very interesting organ that it develops only during the pregnancy, and its role is like a gatekeeper, that it will allow the good things to pass, like again oxygen, carbon dioxide, nutrients, glucose, etc. to pass, and it will stop the large molecules, immune cells, etc., which are toxic molecules, which are not good for the baby or the fetus. So it is a gatekeeper. That is, so well, so good. The problem here is that now, I even did not know it before actually starting off this project, that we have so many different medicines and drugs are available in the market, right? As I already said, those drugs came to the market after a stringent clinical trial. But for ethical reasons, clinical trials are not done in pregnant women for obvious ethical reason. But what is the outcome of that? Outcome of that is that out of all those medicines, only for 20 medicines, 20 medicines, we know that out of those 20, 12 are safe, 8 are unsafe. So I know that 12 medicine can be given to the pregnant woman, 8 cannot be given to the pregnant woman. Rest of the medicines, we even don't know. So as a result, when a woman get pregnant, the clinicians stop many different medicines. So even if the medicine exists, the clinicians cannot administer that. And in fact, 2-3 of the very major such drugs, for example, for epilepsy, which you should not stop, but clinicians need to stop that during the pregnancy because the safety we don't know. TB drug again cannot be discontinued, but we don't know whether that is safe for the baby or not. Sickle cell anemia, again, those drugs, the clinician needs to stop if a woman gets pregnant. So essentially, we don't know that majority of our drugs are safe or not. That's why we cannot administer. So here we are trying to develop a system where we would, so what we are doing, we are having one layer of endothelial cells mimicking the fetus site, one layer of epithelial cells mimicking the mother site, and this epithelial cell are, so sensitization is done, which happens during the pregnancy, and then we check that what passes and what does not. And then we are, so just to give you one glimpse of the example here, that you can, so here we are looking at a dextran dextran, which is a high molecular weight. So when you don't have the cells, just through the membrane, the dextran can pass very easily. But when you have these placenta on chip, these two cell layers in place, you can see that the concentration in the fetal chamber is not increasing, meaning that the dextran is not passing. So it kind of tells us that, okay, dextran is a large molecule, it cannot pass through the placental barrier. On the other hand, if I use the glucose, I will see that the glucose, because glucose passes not as a simple diffusion, but an assisted transport via GLUT receptors. That the GLUT receptors here accept the glucose and then pass it to the next layer. So it is an assisted transport. So here we can see that when the cell is not there, it just increases in the fetal chamber just by diffusion. But when the cells are there, unlike FIT-C, which could not pass through, it is passing through, that it is increasing, but in a controlled manner compared to no cell. And when it reaches to a high level, again there is a control and it goes down. So that is a beauty of the whole system, is that we can capture some of the interesting functionality of an organ. So very broadly, if I categorize the various organ on chip, kind of they come under, first with the tissue barrier function. So like my lung, kidney, placenta, all of these come under, or the blood-brain barrier, all these come as a barrier function. There we generally have one membrane. One side I will have one cell layer, another side I will have another cell layer. Generally in our body it is epithelial endothelial combination, so generally that is what we make. And then we try to see that transport of drugs, transport of biomolecules, whether virus, microbes can pass or not. That actually reminds me that during COVID, that the Delta virus, that was so fatal because it could go to the lower respiratory tract. Whereas Omicron was not that bad because it was limited to the upper respiratory tract. That difference was actually shown and recapitulated, actually discovered, I will say, using the respiratory tract on chip model. So that is the kind of strength of these models. So this is the tissue barrier function. Then I have another section of the organ on chip that I can call as a flow, where we would like to create, say, the blood vessels. We would like to create the atherosclerosis model, various cardiovascular disease models there. Then some model I can put as a stretching or pulsation, like our lung, gut, heart, where things go to a sinusoidal kind of behavior. That, and you can also see that, say, for example, lung can come under barrier function as well as the stretching. So this classification is not a very hard boundary classification, but a broadly to give you an idea what different types of organ on chip one can make. And then finally some cancer on chip or tumor on chip, where you would like to see the migration, invasion, etc. So fine, we have made the organ on chip. So what? So what next? So this is just some of the very recent journal articles. There are many more, and these are not even representative. I just put it to give you an idea of various different things that the the organ on chip can do. For example, there are this liver on chip that is looking into that, as I told at the very beginning, right, that when I take a new drug, the liver toxicity may depend on whether I'm testing it on mouse or pig or human, because there is a species to species variation. What this work shows, that this species to species variation of drug toxicity, that you can recapitulate using a liver on chip platform. Here what it shows is that it is an integrated platform. Here they are putting gut and liver. So if I take a medicine, oral medicine, it first will go through my gut and then liver. So that they are trying to put together to look at non-alcoholic fatty liver diseases or NAFLD. And then as I was talking about that, a lung on chip or a human airway on chip, which can be used and which actually has been used to recapitulate the first viral infection and then to check that antiviral candidates and the prophylactics. So I am just giving you 3 examples where the field is going. And in fact, now whether it is US FDA or European FDA and the drug regulatory bodies or Indian, Japan, and Netherlands, everyone is talking about to reduce the animal testing. So reduce the animal testing, one point obviously from the animal cruelty point of view, but from the other side, people have started to realize that we are using this model, but it is not a very good model. So whole idea is that to reduce the model to reduce the animal model in the drug testing and to replace that with the various microphysiological models or microphysiological system, MPS, and under that MPS, umbrella, organ on chip is one of that. So this is just it is not an exhaustive list, just a list of various different organ on chip that people are making. In our lab, we are making retina on chip, skin on chip, blood-brain barrier on chip, tumor on chip, and placenta on chip. And then because now this field is growing, there are many different companies which are coming up, commercial entities, which will give you these various different types of organ on chip models, just off the shelf. You can take it, you don't need to develop. You just take it and you use it. But they are super costly at this moment, and particularly for India, for our kind of country, and we have to import it, so it is like exorbitantly, costly for us. Unfortunately, we don't have a single Indian company so far which is venturing into this domain. There are a few, who are, I should not say single, there are companies which are developing MPS or using MPS, but unlike, Emulate or Mimitas, that there is no company which is ready to sell a product. That if I want a lung on chip model and I will call a company, okay, I need a lung on chip model, can you give it? Unfortunately, there is none. So if there is anyone in the audience who is willing to start a startup or have this entrepreneurial instinct, I think this is a field to look into. So fine. So far we have organ on chip. What comes next? So next, the idea is that to create a human on chip where you will put various organ on chip on the same platform, so that, for example, if I want to mimic a oral drug, it will first go through the gut on chip, then it will go through the liver. After the metabolism, the metabolites will go to the kidney, and we will see that how things are getting excreted, how it is getting deposited in bone, whether it is getting deposited in lung, whether it is crossing the blood-brain barrier or not. All of these various different organ on chip that I have kind of listed here. Vision is to put everything together to get a systemic response of any drug that we could study. But as I have already told you, it's a developing field. We are still struggling to make one single organ on chip, really good enough. So as just give one example, like we have put gut and liver together. So we are far away from realizing this goal, but the global community is going towards this goal. So I will skip this slide. So essentially the organ on chip is, other than all of the good things that I have talked about, that reducing the time of that drug discovery, reducing the cost, improving the predictability, etc. It is also an animal friendly approach because this will reduce the animal use for the clinical and the biomedical research. So with this, I will stop here, but let me thank everyone. So this is my present team in my lab, and I must thank all my collaborators because by training, I am a chemical engineer. I am not a biologist. I am not a pharmacist. So I don't have that background. So I strongly believe in this proverb that if you want to go fast, go alone. But if you want to go far, go together. So I believe in going together, and I have these fantastic collaborators. So Deepak is a reproductive biology specialist. Projekta is a pharmacist by training. Shilpi is a cancer biologist, and Devjani is microfluidic expert. So with them, we are trying to develop various different organ on chip that I have already mentioned. All our research, all our learning comes from obviously from the funding support from various different funding agency. And when I say the funding agency, it is essentially the citizens of India. So that brings to me back to where I started, is that we all are using the money that comes from even a thelawala on the roadside. So being a scientist, it is also our responsibility. If I am taking your money to do my research, it is also my responsibility to let you know what I am doing using your money.
Yes.